Section 18.2, Development and Organogenesis. By the end of this section, you will be able to explain how the embryo forms from the zygote, discuss the role of cleavage and gastrulation in animal development, and describe organogenesis. The process by which an organism develops from a single-celled zygote to a multicellular organism is complex and well-regulated. The regulation occurs through signaling between cells and tissues and responses in the form of differential gene expression. Early embryonic development. Fertilization is the process in which gametes, an egg and sperm, fuse to form a zygote. To ensure that the offspring has only one complete diploid set of chromosomes, only one sperm must fuse with one egg. In mammals, a layer called the zona pellucida protects the egg. At the tip of the head of a sperm cell is a structure called a lysosome, or like a lysosome, called the acrosome, which contains enzymes. When a sperm binds to the zona pellucida, a series of events, called the acrosomal reactions, takes place. These reactions, involving enzymes from the acrosome, allow the sperm plasma membrane to fuse with the egg plasma membrane and permit the sperm nucleus to transfer into the ovum. The nuclear membranes of the egg and sperm break down, and the two haploid nuclei fuse to form a diploid nucleus or genome. Figure 18.8. Fertilization is the process in which sperm and egg fuse to form a zygote. To ensure successful development, organisms must ensure that no more than one sperm fertilizes the egg. Ernest Everett Just demonstrated several mechanisms at work at different rates. The fast reaction included what Just called a wave of negativity, in which the membrane potential of the egg cell altered quickly. Then the slow block involved changing the membrane structure itself. The acrosomal reactions take place at one location of the egg membrane. The egg releases proteins in other locations to prevent other sperm from fusing with the egg. The development of multicellular organisms begins from this single-celled zygote, which undergoes rapid cell division called cleavage to form a hollow ball of cells called a blastula. Figure 18.9. A. During cleavage, the zygote rapidly divides into multiple cells. B. The cells rearrange themselves to form a hollow ball called the blastula. In mammals, the blastula forms the blastocyst in the next stage of development. Here, the cells in the blastula arrange themselves in two layers, the inner cell mass and an outer cell layer called the trophoblast. The inner cell mass will go on to form the embryo. The trophoblast secretes enzymes that allow implantation of the blastocyst into the endometrium of the uterus. The trophoblast will continue to the placenta and nourish the embryo. The trophoblast will contribute to the placenta and nourish the embryo. The cells in the blastula then rearrange themselves spatially to form three layers of cells. This process is called gastrulation. During gastrulation, the blastula moves in on itself and cells migrate to form the three layers of cells in a structure. The gastrula, with a hollow space that will become the digestive tract. Each of the layers of cells is called a germ layer and will differentiate into different organ systems. Figure 18.10. Gastrulation is the process wherein the cells in the blastula rearrange themselves to form the germ layers. The three germ layers are the endoderm, the ectoderm, and the mesoderm. Cells in each germ layer differentiate into tissues and embryonic organs. The ectoderm gives rise to the nervous system and the epidermis, among other tissues. The mesoderm gives rise to the muscle cells and connective tissues in the body. The endoderm gives rise to the gut and many internal organs. Organogenesis. Gastrulation leads to the formation of the three germ layers that give rise, during further development, to the different organs in the animal body. This process is called organogenesis. Organs develop from the germ layers through the process of differentiation. During differentiation, the embryonic stem cells express specific sets of genes that will determine their ultimate cell type. For example, some cells in the ectoderm will express the genes specific to skin cells. As a result, these cells will take on the shape and characteristics of epidermal cells. The process of differentiation is regulated by location-specific chemical signals from the cell's embryonic environment that sets in play a cascade of events that regulates gene expression.